So I give you this lecture in two parts. The first part will be done as a flip classroom, done online in terms of being a video, while the second part will be done physically in the lecture room itself. So the first part, which will be in a video form, and it will cover the carbohydrate of physiological significance. So the physiological significance part will be covered, some 40 plus slides by using this video itself. And then the digestion and absorption, while role of dietary fibers covered in the classroom. Of course, it is also accompanied, the video will be accompanied with some quiz questions for you to answer and therefore improve on your understanding of the flipped classroom. Right. So, some overview about carbohydrates. It's the most abundant biomolecule on Earth, especially one of its member, that is cellulose, is found everywhere as woody plants, and it's impossible to digest it by many organisms such as humans, but it is possible to be digested by ruminant animals such as cows. And the compound itself, it is CHO having one ratio of 1 to 1, and shown here the ratio. So N decides what will be the carbon number and also oxygen number, will multiplication of 2 for the hydrogen. So example given for glucose is C6H12O6. And because carbohydrate actually has water molecule, at one point the name of carbohydrate came about because of its presence of water in carbon, and therefore carbohydrate. Uh, Basically, or importantly, carbohydrate is for energy source and also storage of energy, for instance, in the form of starch or glycogen. And having the hexoses such as glucose, mostly glucose as a sugar, blood sugar, they call it, because most abundantly glucose is used in the body, be oxidized to simpler forms such as carbon dioxide and water at the end. And in the process, there will be release of energy which can be captured in the formation of ATP. Who has mentioned here carbohydrates such as the glucose, simpler monosaccharides, are source of chemical energy to be oxidized to get ATP. But it's also possible excess of glucose is stored starch in the plants, while glycogen in liver and muscle of a human being, and therefore when required can be reversed to get glucose back. And in terms of structural components, cellulose, this is the one, the woody plants, cannot be digested and used by humans, but nevertheless can be used as building uh, parts in building houses, for instance, the woody plants. Chitin is found in the exoskeletal or the body of insects, and chitin is a modified polymer of glucose. Okay. Also have other roles has been highlighted here. The three major size classes of carbohydrate, there are many ways to actually classify carbohydrates. This is one way, not necessarily that you must think this is the only way. So here, monosaccharides, they are simple sugars, also referred as monomer, and the monomers can be together to become disaccharide, two monomers such as glucose and glucose, and then you get maltose. Galactose and fructose also can be combined. For instance, glucose and fructose can be combined to be sucrose. So that's disaccharide. Looks like oligosaccharide in this classification referred to disaccharide, trisaccharide, tetrasaccharide, and so on. It is 2 to 10, but anything more than 20 is known as polysaccharide. Under polysaccharide, there is cellulose, glycogen, starch. Here we get a lot of information about monosaccharide. So you can see it's simple sugar, and then individual sugar molecule refers to having no glycosidic bond. If it having one single glycosidic bond, then it's disaccharide. And then classified on basis of number of carbon atom. It's not like, so many number of carbon. It's only 3 to 6. So if it is 3, then it's triose. Uh, and if it's 4, it's tetrose. And then so on. 6 is hexose. And in the linear open chain form, yeah, the monosaccharide can be acyclic or cyclic. If it is acyclic, then it's called as open chain. And then it is able to connect at the carbonyl group to become cyclic. We will cover that later. So this is the one I said, 3 carbon, it is triose, while 4 carbon and so on, it is tetrose and goes on. Okay. Right. So this is how you line up the acyclic or no ring structure. For instance, 3 carbon is glyceraldehyde, and you put the carbonyl group at the top, very above, and then line up all the other carbon, and then you put H or OH on either side. And this is referred as aldose as opposed to another one, ketose, because at the uppermost carbon, other than carbonyl group, there is no other R group. If there is a R group on both ends, then that is a ketose, which we will see in the next slide. And then you can see also here, you know that this structure is a aldose, and also triose means got 3 carbon. So more on the monosaccharide, we learn a lot when it comes to carbohydrate lecture. We learn actually a lot of chemistry as well. So that makes it a bit tough. But just hang in there and understand. Therefore, you get used to all the chemistry part of carbohydrate lecture. So, yeah, you have two types of sugar, could be aldoses and also ketoses, based on what is referred as having the aldehyde group, the carbonyl group, and attached to hydrogen rather than another R group. If it is attached on both sides, the carbonyl group, with R groups, either R1 or R2 or just R or same type, then it's a ketosis. So here already given the definition of what is aldoses, while here on the ketosis. An example given here where glyceraldehyde is aldose sugar, where you can see hydrogen is actually connected to the carbonyl group, while here the carbonyl group on both sides there is a R group. Although for this one the R group is similar, but it's still a ketose sugar. And examples are given fructose is quite the popular example for ketosis, while glucose is the popular example for aldosis. Next few slides, we actually look at this more deeper sense of chemistry. So this one called stereochemistry of carbohydrate. So let's look at the following slide. This is on monosaccharide, where the aldotriose, I already referred to you what is the meaning of it. And then if you look at it, it can be represented, since it's acyclic, no ring, because only got three carbon, how to put it into ring. Four also, I think, cannot. Five, six can be put into ring structure, which we will look into that representation. But this is called Fischer projection, where shown here, one carbon attached to hydrogen and OH, and then the carbonyl will always be put on top, and then CH2OH. And this, from being called isomer, they can also be called enantiomer. Enantiomer means they are similar in terms of number of carbon, hydrogen, oxygen. They only differ in the orientation of the OH. The OH facing towards the right is referred as D, while the isomer which has the OH toward the left is referred as L isomer. On top of it, this also can be called enantiomer. So enantiomer refers to a pair of structure that are mirror image of each other. So as shown here, these two are enantiomer. Just bear in mind, it will be better understood if there is more of this carbon before CH2OH. If there's another carbon here, then enantiomer can be understood much better. You just learn about enantiomer and then isomer, then comes epimer. Epimer just refers to only one area of the structure to be different. And one good example normally used is glucose and galactose. Both are isomers, except only this area, this region, you can see OH is to the right and hydrogen on the left for glucose, while the galactose, OH to the left and hydrogen to the right. And that is an epimer where everything else is same, only at this area. Strangely, only so little difference between these two, glucose and galactose. Nevertheless, biochemically, they differ in where they come into the biochemical pathway. Just slight difference can make a huge functionality difference in biochemical pathway. More terminologies related to chemistry of monosaccharide, therefore chemistry of carbohydrate. Now we learn something about chiral. Chiral also referred as stereogenic center. Stereogenic center or chiral refers to this carbon here, the middle carbon, and all the members attached to it are different. They have no similarities. That means there is no like hydrogen here, also hydrogen here, or OH here and also OH there. If OH and OH is found left and right, then they are not chiral. And then if you put a mirror, they will be, twins. So if they are not twins, then they are considered as isomers. Okay. So to demonstrate, let's look at this structure. If you superimpose another structure with the OH on the right, you will see they are not really mirror image. And therefore, that is chiral or stereogenic center. The importance of stereogenic center or chiral is in deciding on how many isomers they can form, and that we will cover in a while. Now, with the understanding of chiral carbon and also stereogenic center, it is possible to determine how many stereoisomers for each chiral carbon. If you have one chiral carbon in that structure, then you will get two stereoisomers. If you have two stereogenic center or chiral carbons, therefore, by using this formula, the n you put 2, it's 2 power of 2, which equivalent to 4. So if there is 2 chiral carbon or stereogenic center, there will be 4 isomers. And if it's 3, it will be 8 isomers. Okay. So shown here for glyceraldehyde, it has one stereogenic center or one chiral carbon. Therefore, the total number of stereoisomer is 2. One is L-glyceraldehyde. Remember, the OH on the right is a D, while the OH on the left is L. So to demonstrate this using 2 power of n, given here glucose as an example. Glucose has actually 4 stereogenic center here, 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 and here, because they all have different molecules or groups attached to them. When they have all 4 different groups attached to them, then they are called chiral carbon. And glucose has 4, then you put 2 power n, 2 power 4 is 16 stereoisomers. So shown here 8, but don't forget, when you flip the OH to the other side by putting a mirror, that will be L-allos. And then similarly, all L-althros when you put a mirror. So apart from showing 8 types of D-glucose, there's also 8 types of L-glucose isomers. In this slide, a number of things going to be described to you. The first thing is about Fisher projection as comparison to Howard projection or Howard structure. Fisher projection is for something of acyclic or straight chain as shown here. So this is 6-carbon glucose where it is now in acyclic structure, open up without a ring. And additionally, you have to realize there's also D-glucose as well as L-glucose. These are enantiomers where it can be OH towards the right, then it's D enantiomer. When the OH is a mirror image and put on the left, then it is L enantiomer. So that's enantiomer for the same location, while isomer is for various other locations such as here and here. And then something to view two-dimensionally, then become three-dimensionally. You can see the wedge bond, dash bond. When it's a wedge bond, this is the wedge bond here, and then this is the dash lines. So the wedge lines is actually towards your view, much closer to you. Just imagine the wedge line is closer to you while the dash line is going away from you. So based on distance, the wedge line is much nearer than the dash line. It's just giving you an impression in Fisher projection. No need to be two-dimensional, can also be three-dimensional. And then you have Howard projection, which is this ring structure, which get connected from the C5 and C1 to become a ring structure. And then, yeah. You can see C5 get connected to C1, become this ring structure, and it is a Howard projection. When it get connected at C5 to C1, two possibility can take place. One is alpha anomer or beta anomer. And shown in the diagram here is alpha anomer because the OH is downwards. And the beta anomer, the OH will be up, uh, uh, upwards. Okay, so whichever projection is dependent on how the connection takes place between C5 and C1, right? Also, another a lot of terminologies to be learned in this one slide itself, also known as Piranos and Furanos. Piranos is a six-membered ring. If you count one, two, three, four, five, and six. So six members required to make this ring structure. Therefore, it is known as Piranos. Fructose normally will have five-membered ring within the ring itself. This is just to give you an idea how the C5 carbon get connected to the C1 and form this anomeric carbon, which OH could be downwards or OH could be upwards. If it's downwards, then it's alpha D-glucopyranose. The previous slide, we learned about Howard projection to be ring structure from acyclic become ring structure. Therefore, it forms that Howard projection. In actual reality, the most what do you call stable structure for glucose is this chair conformation because it has very low energy tension. So this is best. It will represent best for glucose rather than Howard projection in the cyclic way. Okay, so actually this is the Howard projection, and the chair projection is less strainful on the bonds energy wise. So why is it called chair conformation? Because it is a chair at the beach. If you can see, you put your head there and the legs there. So hopefully you get to see the chair structure based on what's been shown here. So we now go and look into monosaccharide, then disaccharide, and then polysaccharide. For monosaccharide, there are three examples given here. That is glucose, galactose, fructose. They are all six carbons. These are the structures, how they look like in the cyclic position or cyclic representation. So, pyranos, referring to it to be having a six-membered ring, and then have a chair conformation. I told you just now, glucose will be best represented as a chair conformation rather than Howard projection. And then for fructose, slightly different because it is a five-membered ring. If you make a count, one, two, three, four, five. So five-membered ring compared to this one having six-membered ring. And then supposedly this does not have a chair conformation compared to glucose and galactose. So, a bit of exercise to recap on what we have learned so far. I supposedly should ask you what is the complete name of the D-glucose. So, glucose is for sure it is pyranose because it's six-membered ring. Therefore, we eliminate the other two answers, C and D. And this is the tough part. A lot of students cannot remember about this anomeric carbon, where the OH could be upwards or downwards. This is upwards, therefore it is beta D-glucopyranose. That's the correct answer for this question. Next question, what is the complete name of D-arabinose? So D-arabinose, if you make a count, one, two, three, four, five-member ring. So let's eliminate the pyranoses, that is C and D. And then what's left is to see whether the OH is downwards or upwards. Since it's downwards, then it is alpha D arabinofuranose is the correct answer. This is the self-study, meaning that you should find out in detail what is the meaning of these terminologies, which we happen to already learn in the slides previously. That is aldose, ketose, furanose, pyranose, and also Fischer projection. I would strongly recommend for you to use AI if it is a bit still difficult to understand based on my description of these terminologies in the previous slides. Right? Howard projection and Fisher projection have huge differences. So find out. And then also chair conformation. We just went through it in a few slides ago. And then monosaccharide as well as anomeric carbon. So I'm not going to go in detail in describing these terminologies, but right, you should figure out on your own. Now we have the other terminology referred as muta-rotation. So what is this muta-rotation? To just get into describing this muta-rotation, you have example of glucose whereby it can be in three physical forms. And for glucose, the amount varies from that of alpha D-glucose, beta D-glucose, and also the acyclic aldehyde. Uh, Based on this carbon here, carbon 5 will get connected to carbon 1, and then whichever cyclic form is dependent on what happens at the end. Okay, but most of the time, 63% will be beta D glucose, while 37% will be alpha D glucose when this structure becomes cyclic. So when you add together 63 plus 37, what is left is very almost 100%. So this acyclic aldehyde is very, very minute or very, very little in concentration compared to the alpha D glucose and beta D glucose. Another thing, alpha D glucose is about the OH downwards. You see this wedge line is actually coming towards your view, like the distance is nearer to you, while this straight line is far away from you. Therefore, the OH is downwards to this structure, and therefore it is alpha anomer, while the OH upwards, therefore this is beta anomer. Right. Before we move on to the next slide, just to make you understand, when they rotate between these three structures, the presence of this trace amount actually play a role in serving as a reducing sugar because of the carbonyl group. Whenever the carbonyl group is present for glucose, it will serve as a reducing sugar when they muta-rotate. So that is actually described in the next slides to come. We already learned about the muta rotation taking place between alpha anomer, beta anomer, and also the acyclic structure, which exposes the carbonyl group, and the carbonyl group can serve as a reducing agent. If the glucose has the carbonyl group, therefore it is reducing sugar. And when exposed to uh, agents or reagents such as toluene reagent, Benedict filling reagents, it will cause reducing or reduction of these reagents, and therefore there will be change in color. For example, when argentum oxide in ammonia, it is Ag2 plus, okay, what they call cation with 2 plus charges. And then when it is reduced by glucose, it will become argentum. So there is a change of color where silver will be formed. In fact, it is become precipitant. So if you see a liquid become precipitant and it's shiny and so therefore it is reducing sugar present when you're exposed with toluene reagent. Similarly, Benedict and Failing reagents got copper. Copper is 2 plus and it's blue in color. When exposed to glucose, when it mutarotate to expose the carbonyl group, it will reduce copper 2 plus to Cu or copper 1 plus, 1 plus ion, and therefore there will be a change of color from blue to brick red solid as shown here. When that happens, you know there is a presence of a reducing sugar. So it is an indicator for reducing sugars such as glucose. We also have here shown the reaction itself when the D-glucose is exposed to this reagent. Actually, they have an additional reagent there, that is bromine. The bromine gas will change into bromine ion, and when it gets reduced by glucose, and there will be a change of color. And not only the reagents undergo reduction, the glucose also undergoes oxidation. So it's reduction and oxidation happening at the same time. Therefore, it's a redox reaction. So you can see the carbonyl group, the acyclic structure of glucose, will actually become oxidized to carboxylic group, and uh, glucose will become gluconic acid. A bit of understanding here in this slide. The reason why the acyclic structure of glucose can come about is because this carbon here is hemiacetal. Okay, so when it's hemiacetal, just like this structure, it can open up and go back and become acyclic. So only hemiacetal can open up to become acyclic and exposing carbonyl group. Similarly, fructose is having hemiketal. The hemiketal can open up and expose its ketose group. So when that happens, it's a reducing sugar. However, there are some molecular structures that do not have hemiacetal, they have acetal. How to know whether it's hemiacetal or acetal? By looking at the carbon, and it's not a chiral. That means it does not have four different groups attached to it. Something is similar. For instance, this carbon has oxygen and also oxygen attached. So it is not a chiral group or stereogenic center. Therefore, it's acetal and it cannot open up to expose the carbonyl group. And eventually, it is known as a non-reducing sugar. And this has been put in the last sentence here. This is a glycoside, acetal, so they are not in equilibrium with any acyclic aldehyde. Only when there is a aldehyde in acyclic structure, it can be a reducing sugar, but because this one does not have acyclic aldehyde, making them non-reducing sugar. Also, you can physically watch under microscope the formation of ozazone crystal. And this is done after you expose the reducing sugars such as glucose under the liquid of phenylhydrazine in acetic acid. Once that happen, there will be this crystal form, ozazone. For different monosaccharide and disaccharide, there will be different structures of crystal when viewed under microscope. As you can see, for glucose, fructose, and mannose, it will give a needle shape. And this is how the needle shape looks like under microscope. While the disaccharide for maltose, it will be sunflower shape as shown here. Looks like a sunflower, right? And then lactose powder puff. This is the powder puff shape. Just now in the slide at the end, it also stated that sucrose is not a reducing sugar. We will revisit that facts later in another slide on disaccharide. Here, important chemical properties as reducing sugar, as we just went through, it can cause changes in Benedict test and can see the brick red solution taking place or precipitation. And then there's also oxidation, where the aldehyde group in glucose will undergo oxidation to carboxylic group. Then it is the N uh, uh, solution is called gluconic acid, no longer glucose. And then terminal alcohol group. So at the bottom of the glucose got CH2OH, and then it becomes COOH. Therefore, it's an acid, glucouronic acid. And then the of both aldehyde and alcohol to glucosaccharide acid, okay, and also galactose to mucic acid. You may want to chat GPT or AI to find out on these two oxidation. Reduction also can take place of the aldehyde or keto group to become alcohol. So glucose to sorbitol, while galactose to dulcitol. Okay, so that means formation of alcohol in the sugar. Dehydration, when treated with concentrated sulfuric acid monosaccharide, such as glucose, will undergo dehydration with elimination of water. Okay. If it's disaccharide, it can actually cleave the bond, but we are talking about the monosaccharide. Esterification is adding of phosphoric acid, so glucose become glucose 6-phosphate. What are physiologically important derivative? Derivatives are modified sugars. Okay, so here we're gonna look into sugar acids, where at the top, carboxylic group is formed. If you recall the structure of glucose, will have at the top CHO or carbonyl group. So once it is oxidized, it will become an acid, and it is known as gluconic acid. And then at the bottom, it will be for glucose, it will be CH2OH, but once it's oxidized, it will become a carboxylic group. Therefore, it is known as D-glucouronic acid. And they serve as conjugating agent. Please find out what is conjugating agent on your own. A derivative or modified sugar, that is amino sugars. Amino sugars are simple. If this is galactose, supposedly here should be OH, but now modified with addition of amino group. Therefore, it is now known as galactose amine. While for the mannose, supposedly here should be OH. It is now replaced by amino group. Okay, it is then known as mannose amine. Okay, so that is amino sugar replacing the OH with amino group. More on the modified sugar, and this one is much more complex modification, and it is mannose that has undergone a lot of modification, as shown here in red, and also the R group is this one, can't even fit there, so it's put on the side. And also the carboxylic group with hydrogen removed, so it has charges. So overall, it has charges, and it is known as NANA or N-acetylneuramic acid. And this belongs under a bigger group known as sialic acid. is an important modified sugar of monosaccharide where it is ribose and also deoxyribose, which is useful to make DNA. Deoxyribose is a sugar useful in making DNA, and ribose is the one useful in making RNA. So it is made into ribose first, and then this OH will be reduced by using NADPH to actually yield hydrogen here, and therefore this is deoxyribose. Before we go into disaccharide, let's finish off by understanding more physiologically important glycosides, more derivatives. So glucose added with vanillin is glucovanillin, and it's a natural substance that imparts vanilla flavor, so the good fragrance for flavoring. Okay. And then we have cardiac glycosides such as digoxin or digoxin and also digitoxin, which stimulate muscle contraction. We also have something obtained from microbe, streptomycin, can become an antibiotic to treat tuberculosis. Then you have iobin, that is able to inhibit these sodium and potassium ATPase and block the active transport of sodium inside. Sure, you already come across this disaccharide in your pre-U days. So it is a monosaccharide linked together by glycosidic bond and less readily broken during transport. It's quite rigid because it's a covalent bond and consists of two monosaccharide. That's why it's called disaccharide. These are examples, glucose with fructose, and then condensation will lead towards formation of sucrose. That's the disaccharide. Information is given on the maltose. Maltose is made up of glucose and also glucose. One is alpha glucose, the other one is beta glucose, and therefore the final glycosidic bond will be alpha 1,4 glycosidic bond. So because of the OH is downwards, so it will be alpha 1,4 glycosidic bond. And then other information, the process of linking it together is referred as condensation. You can also hydrolyze this glycosidic bond by exposing to strong acid. And also don't forget your digestive system with specific enzyme that can cleave this. Okay. And another important message that you have to also understand, uh, this is acetal, therefore it cannot mutarotate, but you still have this carbon being hemiacetal. Okay, there is an L there. So once it mutarotate to expose the carbonyl group, therefore it is able to become a reducing sugar. So on a whole, maltose is a reducing sugar because there is availability of a hemiacetal carbon. The information I already provided in a previous slide. So this is how maltose look like. And as I said, alpha 1,4 glycosidic bond. Maltose is an intermediate product in starch digestion. So when you break down this bond, you will get glucose. And it is sweet, and therefore it is used in candies, okay, in candy making. And you can extract it from sweet potato, syrup, barley malt, sugar. More information, which I already given on the slides earlier. So again, it's just talking about the alpha 1,4 linkages, which is a glycosidic bond. Because of the OH is downwards, it will form these alpha 1,4 linkages. The rest you go through it on your own. Not much information that I have not already described earlier. One disaccharide that actually stands out is because sucrose is not reducing sugar. Why? Because the glucose and fructose that forms the glycosidic bond is alpha 1 and beta 2 of fructose. And these two carbon of glucose and fructose are the crucial carbon that supposedly mutarotate for glucose and fructose. And since for sucrose, they are tied up, so here tied up in the glycosidic bond, they cannot end up mutarotating to expose the glucose carbonyl group and also the fructose ketose group. And therefore, they, as a whole, sucrose is not a reducing sugar. Nevertheless, if the bond is broken down, exposing glucose and fructose, they are reducing sugar. So if it's not possible to break this bond, on a whole, sucrose is known as non-reducing sugar. And also shown here, invert sugar. It's also known as invert sugar because uh, sucrose will invert optical light differently compared to its the members that make up sucrose, that is fructose and glucose. They invert light differently, and sucrose invert in a opposite manner. Now move our attention to lactose. This one, lactose, I'm sure you heard of lactose intolerance, so therefore we're going to visit that understanding as well. But firstly, lactose is made of glucose and galactose, and it's found in milk. And that's why certain people cannot digest or break down this lactose to glucose galactose. Will be having problems, and therefore they are known as lactose intolerant individual, because this lactase is the only enzyme can break or cleave this link of beta 1,4 glycosidic bond that you can see here. Okay, it's called beta because OH is upwards. So since lactase normally quite active when you are a child, but when you grow up, some people lose this lactase, and that's referred in the next slide. So when the intestinal tract do not produce lactase, normally in individuals of African and Asian origins, they lose these lactase capabilities when they become adults. And this is a problem where when they do consume milk, the lactose will be undigested, and it will lead from the digestive tract to the colon. And at the colon, fermentation will take place by bacteria present in the colon, and it will produce all these products which are giving problems to the individual. These carbon dioxide, hydrogen, and also organic acid will cause painful digestive upset, and also could lead to diarrhea for some individual who consume milk, and, and then they are lactose intolerant. Can be overcome by including lactase pills. You can buy lactase pills such as lactate, or you can have the milk to be treated with lactase first and then put into carton and sold to these lactose intolerant individuals. No point people who are normal to drink that milk because they have no issues. They have lactase, no problem in presence of lactase in their intestinal tract. Move on to something of in between polysaccharide and also disaccharide. And, as you can see, 3 to 10 monosaccharide has been loosely termed as oligosaccharide. And they form linkages with amino acid and lipid. So they can be conjugated from carbohydrate with protein to become glycoprotein, also with lipid to be glycolipid, and have a role in being antigenic uh, during blood grouping, ABO. So has something related to the blood groupings. And you also have for the possibility of the blood to circulate without getting attached or getting damaged in the what do you call the uh, vessels, or the lumen of the blood vessels. So they are actually coated with glycoporins, which is having protein. So they are glycoprotein on the membrane of the red blood cell. Therefore, they can circulate easily without getting damaged and also smooth flow. Okay. Also present is the oligosaccharide present in the ApoB, in the receptor, polyprotein of the lipoprotein, LDL, VLDL, they have this ApoB and turns out to be they are oligosaccharides. We have to conclude on oligosaccharide, two example oligosaccharide. One is raffinose. This is a trisaccharide and having trisaccharide of glucose, galactose, fructose. And this is present in sugar beets, so they are known to be sweet. Also cottonseed and many other many cereals. Okay. And then you have stachyose, four sugar moieties, and then having two alpha D galactose. So four, two is alpha D galactose, and then one is glucose, and one more is fructose. Found abundantly in legumes. Move on to polysaccharide, the last classification, and then we look into modified sugar. Uh, and so this one, polysaccharide, is the most abundant because one of its members, cellulose, is found in all the woody plants. It cannot be digested by many organisms, only digested by ruminant animals because they have microbes in their gut that has ability to digest cellulose, but not others. Long polymers, as we will see for glycogen, cellulose, and also starch. So they're also known as glycan or complex carbohydrate. And then they differ from one to the other based on the monosaccharide, how long is the chain, the type of bond linking the units. It could be alpha 1,4 or beta 1,4, and also have branching of alpha 1,6, and also the branching as shown below. Right here to describe on polysaccharide, some could be homopolysaccharide, some could be branched, some could be unbranched. So if it's all the same type of sugar like glucose, it's been put as yellow, okay, or straight chain is unbranched. If it is having branching, then it's branch type of homopolysaccharide. While the heteropolysaccharide having two different type of sugars, monosaccharide has been indicated in blue and pink there, and also they are also unbranched and also branched, explained here further. So here a comparison between two types of polysaccharide, which is starch and also glycogen. A lot of information given here, you may read it, but I just want to highlight a few. Starch and glycogen are both made of glucose. One has more branching while the starch has less branching. Starch is made of amylose 20% and then 80% of amylopectin. The amylopectin is the one got branching that looks like glycogen, while the amylose is straight chain. Okay, and glycogen is stored in the liver and skeletal muscle, while starch in plants. Okay. And yeah, that's the amount of information I can give. The rest you read and understand on your own. Right. So this is on starch in plants, and they have amylose, which are no branches, which actually looks helical. Amylopectin has branches and looks actually similar to glycogen. Only difference is in glycogen has more branching, while amylopectin less branching. And shown here, the glycogen granules could be either myocyte, muscle, or hepatocyte, which is liver. Right. A nice comparison in table form on starch, which is made up of amylose and amylopectin, 20%, 80%, and they are found in plants. And then you also have the other member of polysaccharide, which is cellulose, also found in plants, but uh, starch will have 1,4 linkages as well as 1,6 branching linkages, but cellulose will just have beta 1,4 linkages. And since it has beta 1,4 linkages, it cannot be hydrolyzed by many organisms that do not have the beta glucosidase enzyme in their stomach. So they are left undigested and just go into becoming feces. So they are very good laxative. Okay. Glycogen um, found in animal tissue. So they have both 1,4 glycosidic bond and also 1,6 branching linkages. And yeah, in the cell, not in the digestive tract, in the cell there is glycogen phosphorylase, which can break down glycogen to release glucose. And starch is for energy, while glycogen is also for energy. While the for cellulose, it is structural part uh, forming cell wall. So here we complete the polysaccharides by having describing about inulin and also dextran. Inulin is a fructan and it is a polymer, and it is actually linked together by beta 2,1 bond and found naturally in garlic and onion. uh, it is not utilized by the body, which is good because it's actually utilized by microbes, bacteria that is found in your gut, where inulin will be firstly converted to prebiotic prebiotics. Uh, Compared to probiotics, prebiotics are more of food for our good microbes in our body, while probiotics is the good bacteria itself. Okay, so look like inulin. If you eat consume onion and garlic in your diet, you are actually giving the good food that it can be converted into, and therefore your gut microflora can flourish. Okay. And then dextran, this one not so good because they form dental plaque. And then look at their structure itself. They are obtained from bacteria and yeast, polysaccharide, and their structure-wise is a bit odd compared to, say, glycogen. For glycogen, alpha 1,6 is actually the branching bond, but for this, it's actually forming the chain itself, while the branching is done by alpha 1,3, 1,2, and also the one that is happening in glycogen is 1,4. So it's a reverse of glycogen altogether. And Dental plaque is formed in your teeth, which is rich in dextrin, okay, by bacteria growing there. So that's not good. What is good about dextrin is also synthetically formed, and this synthetic dextrin is known as Cefadex and useful in chromatography method to separate protein based on size and charges. Finally, come to mucopolysaccharides, also known as glycoaminoglycan. Okay, I know it more familiar with glycoaminoglycan or GAG. Okay, but nevertheless, mucopolysaccharide. These are long chain polysaccharide that present in the mucus and fluid around the joints. So those liquid part of the joint is uh, made up of glycoaminoglycan. And also connective tissues such as ligament, tendon can be then formed as well by using these three ingredients, water, collagen, and proteoglycan, and a compound consisting of protein bonded to mucopolysaccharide group. So the proteoglycan is the one having the GAG, and combined they form connective tissue. So these are major categories of GAG: hyaluronic acid, heparin, heparan sulfate, chondroitin sulfate, dermatan sulfate, quite a lot of types. But they all have the amino group distinguish them from sugar alone. So these GAG needed to be modified by certain enzyme before they become hardened and solidify to form connective tissue. Although the main ingredients, water, collagen, and proteoglycan, looks like need certain other enzymes to solidify them to become connective tissue at the end. One should consume balanced diet in carbohydrate as well as protein for the healthy formation of connective tissue, since they are made of protein and also carbohydrate. We now focus on examples of mucopolysaccharide, hyaluronic acid. Your skin is very dry. That means it's lacking of this hyaluronic acid. You can always buy the cream that has hyaluronic acid and apply on the skin to create moisture. And then you also have heparin as well as heparin sulfate, heparin with connection with sulfur. These are anticoagulant. So during surgery, you don't want blood to clot. So applying them, or giving this heparin or heparin sulfate will be helpful to conduct the surgery without disturbance of blood clotting. What else? We also have the mutton sulfate. That means as sulfur, as shown here, sulfur there. And this is the repeating unit of the GAG for the mutton sulfate. Play a role in coagulation, cardiovascular. So all is useful, not bad for causing cardiovascular. Actually useful for these roles. Chondroitin sulfate. This one of the building blocks of cartilage. When the cartilage get worn off, especially elderly people, they have to do a surgery to put metal as the joint, so that it will not, their cartilage already worn off. They can bone and bone rub each other, can be painful. So have to replace the bone. But some people try to avoid surgery by taking this supplement, chondroitin sulfate, although the evidence are mixed, not conclusive yet, but it's been known to help to reform the worn of cartilage. And one more, keratin sulfate. This keratin sulfate, highly hydrated molecule, which in joints can act as a cushion to absorb mechanical shock. And this is how it looks like. The N can be a big number, but since it's keratin sulfate, and therefore has a sulfur in it. So each one of the mucopolysaccharide, they are found in nature and also been used to be applied for healthcare as well.